university's protocol is different. Every university's protocol, submission, modality, procedure is different. It is not same, but I give an overall view. You can titrate according to your university's regulations. So it is not necessary that you have to follow one rule. Based upon your university's guidelines, prepare your protocol. But in case if you think that there is something missing here, you will be able to patch it up. But you can always extrapolate the findings from one aspect to another aspect, because most of the things I may be giving examples taken from pharmacological studies, because I've gone to over about hundreds and thousands of protocols in various subjects, but I may not have retained all those things with me. So whatever the generic ones that come to my mind, which I'm teaching for the last 20 years in this line, with nothing to do with the FDA work. These are all extra work, which we have developed myself and Dr. Balkumar and my team have developed this one for over several years, written 2 books on this theme itself, which I'll be showing at the end of this talk. So you can just grasp it. It's a very wide thing. It keeps on changing. It's a very dynamic thing. But however, the fundamental principles and concepts, they remain same. They don't change it. So on this basis, I feel that each and every candidate or the staff members or the guides who are attending this one, they can draw some stimulus from our talk and incorporate those things in their guidance, curriculum, and the teaching one. Okay. Thank you very much. I've just given this basics of writing protocol. I'm not going over everything about it. How do you come up with the idea? Because already you have come up with the idea. Already you have written your protocol. Your thesis title already been given by your guide. You've gone through the literature search. You build up your protocol and ready for submission. Assuming this one, I am giving advanced features, not the basic features. Usually when I come in December, I talk from the scratch, which will go over 2 hours. But here, I start from the middle, assuming that you already have gone through all the steps. Okay? So still I brush up for a few minutes what we can accomplish in our talk. Okay? This is the outline of my talk. I'll be talking about that. What are the pillars in writing your protocol? That is the number 1 is topic of research. Number 2 is research question, hypothesis, aims, and objectives. These are the 4 things you might have already done it in your protocol, because already title of that is the topic of your research already discussed it with your guide, and then you might have developed research questions. Based on you might have already coined a hypothesis, what you want to demonstrate, and then you would have listed the objectives, and based upon this one you would have written your materials and methods, study design and everything. And then at the end, need of the research, why you should do the work. A lot of different terms are coined by each university. Somebody writes rationale for the research, somebody writes significance of the research, need for the research, or need for the research. So many terms are there, but they're all meaning same thing. Extrapolation is very important. Then I'll summarize my talk. You know, writing anything is a very tedious one. You can always talk, blah, 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 blah. You can keep on talking anything and everything, but putting in a proper fashion, grammatically correct, with a good style is very important. And you know that when you attend to the movies, you've seen that the screenplay is so fantastically, the screenplay writers, they are paid thousands of rupees, lakhs of rupees they pay for them. They write so nice one. Similarly, you are supposed to put it together like this way. And many people do not write very nicely. That's why it's boring for them to write also, boring for us to read also. So make it a habit to write very nice. That's why good science is exciting, thought-provoking, interesting reading, and will improve the investigator's chance of getting funded. Funded means in this case, I'm using this one, that is your review committee will agree with your proposal to go ahead with your work based upon the solid thing what you propose here. All proposal starts with the innovative ideas, which you already discussed with your guides and got that research topic. And based upon this one, you have got a number of questions you posed it yourself, and how you are able to answer those questions in your methodology. And based on research questions, you develop a hypothesis, what you are going to demonstrate in your work. And protocols do not have clear hypotheses are unlikely to be viewed favorably by your committee, or if you're applying for a small grants for your work. For example, the students are applying for AGCPA grants. In about 8 grants we have, and how you can impress upon us with your writing style. And also aims and objectives are based upon hypothesis and the question, which I'll be showing with 3 examples in each category. Same examples I'll be showing, so they can correlate how a topic can be divided into questions, divided into hypotheses, objectives, and then going towards the study design, which Dr. Balkumar will take over from me. And what is the need for the study? That is the benefits, significance, which we'll be discussing in the next 45 minutes or so. You know, this theoretical, what I'm talking is all theoretical aspects. That is the anatomy of the research. That means you write, put everything on the paper. About 5 to 10 pages is your protocol. There may be some university limitations may be there. Then you start your work. That is the empirical part. So we talk only about the conceptualizing the topic and then writing the theoretical aspects. Then you proceed with the empirical study. So you see that whole scheme, it should revolve around you and your guide with a research statement given by your mentor, your guide, and then consulting the review of literature. You would already done it, and you got the research topic. You put the questions, hypothesis, objectives one, write the need for the study, rational significance, and here it completes your thinking concepts. Then you write the methods, design based upon the objectives only here, and then you begin your studies. Studies, we're talking about this much part today, okay? And both myself and Dr. Balkumar will be discussing only these aspects. Before you begin your research, you got a plan of study, that is research protocol, and you have got a guide, and you have working in a laboratory situation. And why do we require it? It is a roadmap for research. That is the main thing about the protocol. It is a structured outline. A well-designed plan should be there. And without the plan, without a protocol, you cannot start any experiments. That's why building protocol is very important. First protocol should come. Then only you start your experiments. Not after the experiment, build up your protocol. Don't go in the reverse way. A strong protocol is very necessary before you begin your studies. This is what the entire thing, so we're talking about the anatomy of research. Okay, protocol part. So number 1 point is research topic. For that, you require background information, review of literature, past 10 years literature is necessary. Not the 25 years taking from Nadkarni and other books, Chopra book. No, recent papers you should consult as recent as 2024 or 2023, late 2023. At least 1 or 2 references should be there from the recent literature. And then you identify a gap, what work is not done by others. Can you fill up that gap? That is very important. And then research questions, which defines the study and framework, hypothesis, objectives 1, and that is the need for the study, significance, innovation, benefits. You can use the different terminologies for this one. Many universities use different terminologies, but the content remains same. So the comprehensive research protocol is a title, introduction you should give, and of introduction, that means a background for your work, including the compounds, what you are using in your studies, or a plant material. Then follow research question, hypothesis, then objectives 1, significance, literature review, write it about 1 or 2 pages, then methodology, study design, which we'll be discussing the 5th point by Dr. Balkumar, then statistical analysis necessary for interpreting this, analyzing these groups, then what is the target, how you accomplish your work in 9 months or 12 months, and what is your timetable, and references should be listed very meticulously. These are the 4 question 1, 2, 3, 4 fundamentals 1. Add the significance, becomes a panchabhuta. Okay. What is research topic? The research topic is the keystone of the entire scientific enterprise. It begins the project, drives the entire study, and is crucial moving the project forward, dictates the remaining elements of the study. And for that one, a strong literature is necessary. That means you should have digested the literature published, work published in the past 10 years. So this is what very much necessary so that you can avoid any duplicate work. Somebody has already done the work, then what is necessary of doing yourself as other work? So that's why you should be careful about uh, coming up with a good work. For that, you require a lot of ideas, a lot of thinking. So creativity, knowledge base, review of literature, close observation, creative thinking, critical thinking, and moving from various points to narrow into a single point of the work, which I'll be talking about when you go into a topic one. And then you finally come to a research topic. You and your guide are the two people who are responsible in getting a correct topic. If topic is wrong, the whole thing, whatever you write, further one is wrong. So be careful when you select from the variety of array of topics, you narrowing down to only one. This is what I said, moving from divergent to convergent. 100 ones reduced to only one. Because of time limit, you cannot work on all the aspects. Only one aspect, one mechanism you take and work. For that, you require first-year ideas. You should not work on any copycat work. That's why a gap in the literature is necessary. You should search in the literature what work has not done by others. And that particular aspect, you should try to grab it and work. So second tier ideas, most of the work what we do in India and many of the colleges is all second tier ideas. Somebody has done the work on A, you will repeat the same thing and using the B, assuming that nobody has done the B, but people would have done it. May not be in your university, may not be in your state, but somewhere else would have people done it. If you go through the literature, you definitely will find somebody has done the work, because there are millions of people working in this world. Everybody will have touched everything. So be careful about always be in this area, first tiered ones. So copycat research, yes, you can do your work, you can always build a thesis, but you cannot publish that work. What is that? That becomes a piece of junk. That's why we wrote in the recently the Nature, that is avoid predatory work. So that is very important. That means ideas should not be plagiarized. It should be the original. Your idea should be original. You may say that, oh, I'm working in a small college and what I can do for it, and my ideas are not. But however, ideas doesn't come whether you are a small college or big college. Ideas come from the literature search. How much in-depth knowledge you have in a particular area, a particular subject. That is more important. Whether you can grasp that particular work and then see that what has not been done. That is known as the niche, the gap in the literature. And intelligence, a little bit gone over it, I think possibly. For any of this one, ideas, as I said, intelligence, environment, creativity, you know that, I know all of you will understand Kannada. ನಾವು ಸ್ಕೂಲಲ್ಲಿ ಇದ್ದಾಗ ಸೋಮೇಶ್ವರ ಶತಕ ಇತ್ತು, a 12th century poet, and he was a very brilliant poet, Hara Hara Chennai Someshwara. So you might have heard about those people who studied little bit Kannada in middle school, and there it was to be very nice one. Poem is to be there. ಕೆಲವೊಮ್ಮೆ ಬಲ್ಲವರಿಂದ ಕಲಿತು, ಕೆಲವೊಮ್ಮೆ ಶಾಸ್ತ್ರಗಳಿಂದ ಕೇಳುತ್ತಮ್, ಕೆಲವೊಮ್ಮೆ ಮಾಳ್ಪರಿಂದ ಕಲಿತು, ಕೆಲವೊಮ್ಮೆ ಸುಜ್ಞಾನದಿಂದ ನೋಡುತ್ತಮ್. He says that there are 6 modes of learning. 6 modes of learning is here. That is from your teacher, from the class, that is mentor input. ಕೆಲವೊಮ್ಮೆ ಬಲ್ಲವರಿಂದ ಕಲಿತು, that is from your teacher you learn in the classroom, in the guidance. ಕೆಲವೊಮ್ಮೆ ಶಾಸ್ತ್ರಗಳಿಂದ ಕಲಿತಿರೋದು, review of literature. See, this is the second aspect. ಕೆಲವೊಮ್ಮೆ yourself, creative thinking, imagination, intelligence, creativity. ಕೆಲವೊಮ್ಮೆ ಸ್ಕೂಲಿಂದ, ಇದರಿಂದ, environment ಇಂದ, from your friends, from your peers, and from your own judgment, from guidance, and with your experienced individuals, and ಕೆಲವೊಮ್ಮೆ ನೋಡುತ್ತಮ್, that is curiosity, observation. So there are 6 modes of learning. This one of the workshops is another mode of learning. That means you're becoming, attending the workshop, and you listen to somebody. What ideas this guy has got it. So that's why there are 6 modes of learning. This is what at that time Someshwara said. Anyway, that is, now I'm coming again back to this one, my scheme. Say, for example, research topic, what I said about divergent. I'm picking up only one, neurodegenerative diseases, which many people do it. Pharmacology people do it, chemistry people do it, pharmacology people do it, even pharmaceutical people, they pick up this one. Neurodegenerative diseases, various formulations, which I, because for last 2 years I attended several presentations coming from both MPharm and PhD students. I learned this one. So neurodegenerative disease is a very wide terminology, and narrowing down to is a dementia, which is an umbrella one concept, and narrowing down is Alzheimer's disease. See, from big one you come to one narrow one, Alzheimer's disease. Your teacher says that, guide, let us work on some neurodegenerative diseases. It's a very big one. And how do you come to this one? Then you may say that let's dementia, or let us go to Alzheimer's disease. Again, in Alzheimer's disease, it is a very big one. There are about more than a dozen mechanisms which operates. For example, your laboratory is very small, doesn't have very good equipment, anything else. You go for a simple acetylcholine inhibition that increase level of acetylcholine to combat that memory loss, cognitive skills. Very simple one. So, okay, I will go for the seventh. See, from here, the big one, you reduce to disease, and then out of this 1 dozen or more, that is the targets, you choose one of the targets, or I may to go to that of inflammatory molecules. So I can have an anti-inflammatory or antioxidants, or I can go for a neurotransmitter release, or I can go for the brain-derived neurotropic factors, so trypomyosin releasing kinase inhibitors B. So that is why you can choose narrowing down to one point. So this is what the meaning of divergent thinking to a convergent thinking, narrowing down to one point. Now, when you got to the particular topic, research topic, you formulate into a question, a series of questions, what you can do it. The same thing, acetylcholinesterase inhibition, which increase the level of acetylcholine. So you get into a number of questions. A good research must begin with a good research question. It should be a thought-provoking question so that you can only answer through experiments, because you'll be getting into a lot of objectives one based upon the research question. It should be a thought-provoking question. The question is a starting point, sets the framework, and will guide the rest of the study design. So it's important to get it right in the beginning itself based upon topic. Once questions are all right, the question can be converted into a statement, hypothesis. Hypothesis is not like right or wrong. No, nothing is right, nothing is wrong. You only demonstrate that according to this one, this is okay. That's all. That is, you first think carefully about what comparisons will be needed to answer the research question which you put it here. That means statistically, test, you prove it. There is a difference between the placebo, a control, to that of a group 2, which contains an active molecule, active pharmaceutical ingredient, a testable drug, which was synthesized. You have synthesized about a dozen compounds. You would like to compare all the dozen. No, it is not possible. So you pick up 2 or 3 good compounds which you think that in SAR they're active ones. Then you compare control versus 2 or 3 molecules. You got so many groups, so many doses. Then how can you compare it? That is the study design, which Dr. Balkumar will be discussing it. When you got multiple groups, how can you focus on this one? Multiple groups means the multiple animals have to be taken. N is equal to increase of much more. Even if you take 6 animals per group, if you got 6 groups, 6 into 6, 36. Or if you got 10, become 60 animals. How do you handle? What about the cost factor? What about the drug infusion? So all these things you should look into the criteria. And this is a question from hypothesis, everything. From there you get into objectives one. From the objectives you go to methods. For each objective, you should have a method 1, 2, 3, 4. Now, this is a question. It's a question based upon the knowledge gap in the literature. And you already, that means what has not been done. You might be reading the paper, you might say that this has not been explored previously by any of the authors. To best of my knowledge, this work has not been done. When they use the word, that means there is a gap in the literature. That's why this particular authors, research investigator would like to explore it. And that one, you will be catching that one. What is the gap? And for that one, you should have a final test. It should be feasible, Again, whatever the gap is, the exists literature doesn't mean that you can work. I showed you about a dozen target sites for Alzheimer's disease. All of them are not possible to work. You choose which one is feasible for you to work. That is known as the feasible. Should be interesting for you and your guide. Should be a novel, innovative, emanating from the gap. Should be ethical consideration should be there. You cannot take a big animals and work on it. And then relevant to the need for the study. Think about that one also. A research question. For example, remember these titles. Topic 1, topic 2, topic 3. I'll give 3 titles. Same thing I repeat for the research question also. Same thing I repeat for the hypothesis also. For objective also. Then you can be able to focus. I don't want to change it because people may think that, okay, this is the title. What research questions you can come across? So if this is the question, hepatoprotective. See, the title is so nice one. Hepatoprotective should keep in your mind. That means it protects the liver from what? From the damage caused by what? Paracetamol. Where? Which animal? Which drug? Gallic acid. So everything is given here. That means you are studying the hepatoprotective effect of the gallic acid, and the damage is caused by paracetamol, and you are using mice. And question of this one. First, you should know that what is paracetamol? Why does it cause? Either any other drug which can induce, or any other chemical can induce liver injury. Yes, carbon tetrachloride. Can I use that one? Or why should I go for only paracetamol? You should think about it. Discuss with your guide literature and how it causes injury. What kind of injury causes by paracetamol versus other compounds like carbon tetrachloride? What is the mechanism of causing liver damage? And what is the histological features when damage causes normal versus the damaged liver and regeneration with the drug. So these things you should pose questions and whether you can assess this one in objectives one and in the method carry forward to this one. And what biochemical parameters indicate liver injury? That is, for example, SGOT, SGPT, alkaline phosphatase. There are about bunch of enzymes are there. Can we estimate in your lab? You do have a know-how. And if there is a molecule gallic acid, how does it protect? That means there should be inflammation. The compound should be anti-inflammatory. Compound should be antioxidant. That means these two have to explore it. So these are the questions you should write down. See, further mechanism we can explore if you pursuing this one, liver injury or damage caused by either carbon tetrachloride or paracetamol, any other chemical, then antioxidant activity you may have to do it. Anti-inflammatory one, apoptosis, cellular death, stimulation, regeneration, this one, detoxification aspect, immune responses. So these are all things should come into your mind. That's why I put all the questions. Now we'll give second example. Nano-formulated ellagic acid. Nano-formulated break. Other one is ellagic acid. That means you got two compounds here. As I said, people, chemistry people, pharmaceutics people are going like this way, which I read from this one, from a pharmaceutics guy. They want to formulate the known compound ellagic acid with a selenium or with a zinc one, so that it can go for a site-specific delivery. That is the case means the dose differs. That means nano-formulated should be more specific and less dose, more efficacious. That means even if you give 10 times less dose than the regular ellagic acid, that is 0.1 and 1, or if you give to 0.01 and 1 milligram per kg dose, whatever dose you give. That means you are comparing these two. Compare means again statistics comes into picture. Two groups are being compared here. The two formulations, regular formulation and nano formulation is supposed to be better. So nobody would have thought about it. So you want to use selenium, for example, or any other things which can carry the molecule. Ameliorates what kind of this in here? Pentylene tetrazole induced convulsions here, seizures, and that you are using in the mice. And possibly this ellagic acid may be modulate the effect through two mechanisms, possibly oxidative stress and inflammatory cytokines, and regeneration of this one. And that one, either because it affects not the liver here, it is the brain because the seizures coming from here. It's the CNS mechanism. So you're estimating everything in the brains of the mice. And why you're choosing the male versus female, that also you have to discuss here. So let us put the questions here. So we know title should be able to give you the overall picture. And if anybody reads, when I go for presentations, listening to the presentation given by number of students in various colleges, when I see the title, immediately I grasp it, what the student is going to do in their protocol, or what has already been done if they're going to present the results. I would have formulated my thinking in my brain by seeing this descriptive title here. That is ellagic acid that has been nano-formulated here, better or more efficient than EA. Better than. It is more efficacious than. So you are using the word, two things. When you use the word better than, more than, that means you are always using a statistical one. This two batch of students are better than the previous batch of students. That means how do you compare it? There should be some parameter should be there to compare between one versus the second one. Does ellagic acid have a neuroprotective effect? Then how do you measure this one? Neuroprotective effect by anti-inflammatory, antioxidant, and what kind of studies demonstrate neuroprotective effect? What are the anti-convergent properties of nanoformulated ellagic acid and the regular ellagic acid? And what distinguishes them? What biochemical parameters correlate the anti-convergent effects? What kind of inflammatory cytokines? Think about IL-6, IL-1, TNF-alpha, there are NF-kappa B. So some of the things should come into your mind. What you can do this one. Think about the third one. So again, remember all the three titles. Again, you will be coming to this title again when I go to the next topic. Anti-Alzheimer activity of leaf extract of this particular material, plant extract, and its nano formulation. See, uses the polyanthus uses leaf extract, regular one, as well as the nano formulated one. So again, you got two. The number of groups increases. Working single ones, you may be having 6 groups because of varying doses. But if you take another one, 6 plus 6, 12 may be there. So in what they're using scopolamine to induce neurodegeneration in the Wistar rats. You may use Sprague-Dalley or Wistar. So mention the particular strain. How does Alzheimer's disease differ from dementia? You must understand the concept, umbrella concept. Dementia is a bigger picture. Then you'll be having a different type of diseases caused by neurodegeneration. What are the various underlying factors contributing to the development of Alzheimer's disease, which I showed you in the beginning slide? There are 12 minimum targets I showed you. What are the signs and symptoms? For example, affects the memory, it affects the cognitive skills, behavior, cognition. Oh, we can measure in our lab because we are doing psychoneuropharmacology, pole trimming method, or maze climbing, this one, or swimming test. So these are some of the things. And cognition skill, learning, yes, we can do in our lab. So these things should come into your picture. Yes, these are feasible for us to do it. What are the hallmarks of Alzheimer's disease? Beta amyloid and tau tangles. Can you do it, estimate it? Oh, possible, it may not be possible in our lab because we have to buy the kit. Kit costs about $300, $400. So you may have to drop this one unless you can afford to have the kit to estimate this beta amyloid plaques accumulation in the brain cells. You have to extract that one. What is the anatomical pathology of Alzheimer's disease? That is the atrophy and also these plaques and tangle depositions. Which biomarker is validity of Alzheimer's disease neuropathology? So these things should be able to come up with all these questions. So once you are ready with the question, then only you can able to go to the next one. That's why understanding a question is often answer. You have to understand. That means the answer is based upon what? Literature review. You should study the literature. What kind of experiments they have done it to know the answer for the question. That is the experiments done. And those experiments are feasible or not. Can you handle yourself with minimum cost, with minimum laboratory equipment? So designing the study completes the answer, which we will be talking later on. Now, hypothesis. After this question, topic, this is a question, we are moving to the third aspect, hypothesis. It is a central component of the research process. Research questions and hypotheses serve as a guide to the study, explaining its purpose and guiding its direction. The research question should be translated or reframed into a workable, and you have to test the hypothesis by statistically, not by experiments. Statistically, you test it using different formulas, which Dr. Balkumar will be discussing. It predicts in advance what is the outcome of your research. Yes, there is a difference between the control group and the treated group at this particular dose. That means varying doses, low dose, mid dose, high dose. Remember, 3 groups comes into picture here. A hypothesis is a specific statement of a predicted relationship between 2 variables. Minimum 2 variables should be there for an hypothesis. 1 variable, no hypothesis, which I'll be talking in the next slide after giving examples. One is a dependent variable, that is measuring a blood pressure. It depends upon the particular model. That's why dependent variable. What is independent? Externally you give pharmacological intervention. You give number of drugs. That is an independent variable. So that is high fat diet or exposure drug. These are all the interventions in the study. Now I come back to the again same 3 examples I'm repeating. Go back to your notes and see what I gave the statement there. Number of research questions I gave. Hepatoprotective and antioxidant effects of gallic acid in paracetamol-induced liver damage hypothesis in liver, in mice. What is the hypothesis? The gallic acid treatment alleviates, decreases the progression of inflammation in paracetamol-induced hepatotoxicity. This is your statement. Now you want to demonstrate, yes, this treatment, this animal having this injury by gallic acid, it decreases. You want to show statistically, because based upon the experiments, you use appropriate statistics to demonstrate your hypothesis is correct. What you predicted in the beginning. What happens if a negative one? Then you can say, yes, gallic acid did not protect. That's okay. Negative answer is fine. Gallic acid did not protect. That's over. Or you can also use this other one. Antioxidant effect of gallic acid relieves the progression of inflammatory mediator TNF alpha. If you estimated any of the 3 molecules, you put here. Inflammatory molecules like IL-6, IL-1, TNF alpha in this paracetamol treated mice. So any one of them you can use it. So think properly when you're rating the hypothesis. Second topic. Nanoformulated ellagic acid ameliorates PTC, induced seizures, conversions in this male mice. This is what I already stated earlier. It should be in your mind. What is hypothesis? It's hypothesized that ellagic acid as well as nanoformulated one, both of them, they have anti-convergent activity and it may modulate seizures of animal models. Or you can say nanoformulated, you can combine both of them, or you can use the different one statement, you can make it. Ellagic acid is more efficacious. Now we are using the more than what? Regular ellagic acid. Then use the statistics to show that, yes, this is by 10-fold, because you've taken a 10-fold, 10 times lesser dose to get the same effect as that of ellagic acid. More efficacious in elevating the experiment-induced seizures of ellagic acid. See the third example. Again, the same thing, I'm repeating the example. Keep in mind, I'm not changing my examples. Anti-Alzheimer activity of the leaf extract of Polyanthus tuberosa and its nanoformulations. Again, the 2 formulations here in this neurodegeneration induced. What is the hypothesis here? This particular plant extract and its nanoformulations. There are 2 formulations here. Both of them inhibit Alzheimer's disease in scopolamine-induced viscera. Or you can say leaf extract from this one, this nanoformulation counteract Alzheimer's, dementia, neurodegeneration. If you want to make a statement like this way, then you have to work on all of them. This becomes a very broad one. So it is not ideal one to use this one. Cut off this whole thing. In Alzheimer's-like disease induced, in scopolamine-induced one. Put a narrow way. Otherwise, the reviewer may say that you are touching a very broad way, though, although you are not worked on this one. So this one, just for demonstration, I put all the things here. So they can narrow down to only one aspect. Be specific in your hypothesis. Don't beat around the bush, giving a very wide one, which you cannot support from your experimental observations and the results. So results should be able to show that your hypothesis is correct. So now I finished all 3 examples here. Now, hypothesis necessary or what? I said 2 groups, I said over 2 variables. All biological research, including discovery science, is hypothesis driven. What all we do is all biological science. So hypothesis is a must. People should include hypothesis in your protocol. Don't forget, I've seen many people do not include hypothesis from topic. They go directly to objectives one, bypassing research question and hypothesis. I don't know why it is so. Many universities do not insist This is what I told. I met many of the university administrators. I told them, why did you do this? Why didn't you include this one? Sir, this is how the professors of each college, they gave input, we put it there. That means fault lies with the particular college or the where this protocol has emanated. They would not have thought about it. So that's why I'm telling all the guides and the professors, please think about it, modifying your protocol submission where you should include the research question and the hypothesis in your protocol structure itself. That is a must. However, not all studies need to be conducted. You can use the word hypothesis if necessary. So students will include. The students will say, no, my hypothesis is not necessary. You can say not available or not necessary because I'm using only one variable, because I'm synthesizing a large number of compounds, or I'm using a single formulation, or I'm using a new methods for estimating the plasma concentration of this drug diphenhydramine, for example. So I don't need an hypothesis. Why you do not need a hypothesis for your study? This is what I given. Some of the things you may not need hypothesis. Otherwise, by and large, all specialties in our pharmacy, they need hypothesis. Now coming to that last point, there is a fourth point. 4 basic structure, 4 corners, 1, 2, 3, 4. To build anything else, you need 4 corners. One is research topic, research question, hypothesis, then there's an objective one. Now use the word aims and objective. People use interchange this one. That is not correct. See, I'll give the definition, why they differ. What are you trying to accomplish? That is the main thing we should ask the question. What you hope to get, and you want to do this, it is a gateway to the study. Without objectives one, you cannot proceed. Your objective is to come and attend this seminar today. So that is your objective. In the research context, writing a protocol or proposal, there is a clear distinction between the aims and objectives. Both are the desired result of the work performed by researcher. However, the entire different concepts are there. Aim refers to the general direction. My aim is to demonstrate that the particular compound is an anticonvulsant. That's it. Stop it there. That means that is the aim. To demonstrate the anticonvulsant effect of a compound, or an anti-Alzheimer activity of a compound, or anti-diabetic nature of the compound. That is your aim. To accomplish this aim, you have several objectives. One. Now you understood? To accomplish that aim, you have several objectives. Aims are general statement that provide direction and identify the intention to act in writing a research grant proposal. It can be chunked into various objectives. Aims can be split into various objectives. So objectives are a subset of aim. Now we understood? So aim is the top one. Above this one is the goal. Then there is the aim, then objectives one. We are working at the objectives part. So the aim of this research, for example, see this definition with an example. The aim of this research is ultimately the mapping of the whole human genome. Our first objective in order to accomplish this aim is to develop methods. So method, based upon the objectives, you develop methods. Okay, oxidative stress, use the word in objective one. Then you develop what are the different oxidative stress parameters like catalase, GSH, or reactive oxygen species. So all those things comes under objectives one. One word you say antioxidant or anti-inflammatory. Then you see number of methods that are there. Our goal is to sequence X chromosome. This is the main goal. This is the bigger one. So goal is a high level. I want to become a scientist goal. You may become, may not become. That is your goal. To accomplish the goal, your aim is to study. Then objective is to go through hard work. So in this way, you get into different aspects. See the definite, I've given the difference between the goals, aims, objectives. You are working at the objective level because it is time bound and fixed duration, and also it is measurable. Because all the parameters, what you put under objectives one, they're all measurable. GSH for antioxidant activity or IL-6 for anti-inflammatory, they are measurable. The decrease or increase, the increase in the inflammation condition, your compound is about to decrease it, that one. So you are measuring it. Okay. So that is the way how you accomplish objectives versus the aims. Now, the purpose of objective is to describe concisely, realistically what the proposed research intend to accomplish, define needed efforts or actions, summarize what the researchers expected to achieve, and specify the scientific questions that the study is designed to answer. So the objective should be smart, should be specific, measurable, achievable in the time frame, realistic, time bound, and should be challenging. That is, they are supposed to answer very nice question. Objectives are grouped as either primary objectives, secondary objectives, or if you don't want to group it, write objective 1, 2, 3, 4. See the example here. Again, you come back to the same or examples. I've already given this earlier for research question, hypothesis. Now I'm going to give, I don't know what happened. Aim of the study to investigate the antioxidant, phytoprotective and hepatoprotective activities of gallic acid, antioxidant activity, anti-inflammatory, liver function enzymes, study plasma, histopathology liver. These are all your objectives one under this title. Something happened here. I don't know what happened. Suddenly it pointer option. This line has appeared here. I have no idea. Do you see this scribble here?
Yes, sir. We but don't save it, sir. It will go off
Okay.
at the end.
Sorry about it.
Yeah.
I don't know why suddenly it happened, that one. So research topic, again, as a third one, anti-Alzheimer activity. What are the objectives one here? What is the aim? To evaluate the antioxidant activity of this one. Same thing with the previous also. Aim I gave to investigate the anti-convulsive lactic acid, then objective 1, 2, 3, 4. This example also I given a aim, and then give objective 1, 2, 3, 4, 5. Now we have to come up with the methods for each one. For example, characterization of anti-inflammatory profile of the extract through the investigation of IS6. One method. I take KNF alpha, second method, third method. So your methods will be method 1, method 2, method 3 for the based upon the objective 2. You got 3 methods, you're supposed to write it here. And again for the antioxidants, you got DPPH, ABTS, free radicals. This one again you have got 2 or 3, whatever you propose for antioxidant determinations. Again here, again you got one method, second method. So your methods increases depending upon the number of objectives one you have to use it. If you want to do histopathology, one more method. What kind of histopathology you are doing it? So study design, which Dr. Balkumar is going to discuss it. So that's why I said study design can be simple because where you got only control and one treatment, or you got advanced where there are more than 2 groups, or complex, more interventions. That is incremental doses. You have to use minimum 3 doses. You'll be using it. Low dose, mid dose, high dose. In this webinar, we wanted to talk about how to decide the dose, what kind of dose, and what is the route of administration, intravenous or intraperitoneal or oral, how, and what is the volume to be given. All these things we wanted to have one more speaker, and unfortunately the speaker is not in the US, has gone to visit India, and I don't want to disturb him. And this is what I told Rajesh Pandey. 1 hour would have been that one where they will discuss about the acute toxicity of the compound, because you will be using only for 1 month is my study, or 3 months in case of diabetic one. If you're talking about diabetes or Alzheimer's, you'll be injecting definitely for 3 months the drug, and you have to do a toxicity test of any of your leaf extract, new one. If it's already established, literature will tell you what dose has to be selected. So based upon the literature, you will come up with the dose, lowest dose, where there should be something you should get it. High dose, not to kill the animal. Then mid dose, demonstrate an effect. So these are a very, very tough job. How to select a dose. 1 hour duration of lecture, we wanted to give to you people how to select dose, because the entire project depends upon the dose, whether to kill the animal or to get the experiment, to answer the query, or no result will become, negative result will become. So 3 possibilities are there: no, yes, and killing the animal. So that's why the dose is very important. This is what we discussed at the FDA. Always preclinical or clinical, what dose to be given to the patients, for example. That's why a lot of debate, lot of discussion we do for months together, we do this one. That is the first in human dose, FIH. So this is which we are missing. But however, assuming that you people are knowledgeable, based upon literature search, you are choosing, you are selecting a correct dose. Correct means approximately good dose as a low. Don't take too low dose like 0.001 milligram to that of 1000 milligram. That is very, very broad doses. You should not choose. So discuss in the literature, read the literature, and whether you want to use arithmetic dose or log dose. Usually logarithmic dose is selected. And remember, this is a very tough job to get all these things done before you put everything into your work. And this is a scheme which I put it. Everything is related. That's why it's onion shape. It is a stacked Venn diagram to show the literature search based upon your creative ideas, creative thinking, divergent thinking, all these things. You get into your topic. From the topic, you get into hypothesis, aims and objectives, matrix method, study design, statistics. All these things I should be able to give you the significance of study. People write pages together for the need for the study or need for the study significance. That is not necessary. You read only a couple of lines of one paragraph. What is the need, which I'll be discussing in the last part, that is significance of the study. That is the fifth major arm of the protocol. That is, it should be very short, not pages together. I read the protocols. I ask people because they write the history of the insulin. For example, they're talking about history of glibenclamide. You don't require all those things to say that what is the significance of the study. Come to the point. So you don't require pages to write it. I don't know why the guides approve that pages together for significance of the study. You should have only one paragraph, which I'll show you here. The current state of the knowledge, you should know that. And what are the gaps the project is intended to fill? There is a gap in the knowledge base, unmet need that poses a problem. But the objective should be linked to this. For example, development of a vaccine for SARS-CoV-2. That is the rationale. And that's why your whole work you're doing it, because your study is focused in developing a vaccine for monkeypox. So monkeypox vaccine. So they can develop this one, or for malaria vaccine, flu vaccine. So the rationale proposed research, why is the experiment being conducted? Justify the need for a proposed research. What makes your research significant? Is the study to confirm, refute? You should answer all these questions. What are the innovations will come out from your study? So you should talk about these things, not the what work others have done it. People give the review of literature in the need for the study. That is wrong, entirely wrong. I've spoken to many students, even our M.Pharm project work for the last 20 years. I told people this is not the way of writing. Still people write it because the previous protocol written like this way, everybody copies that one style and they do it. So I always teach them, but people don't listen to it. They do their own mistakes. So this is experimental methodology. You'll be having the plan of study, design, analysis, materials, methods, instrumentation, data recording, difficulties and limitations, ethical issues, and the timeline. The whole thing comes under experimental methodology. So final research protocol, how does it look? Problem statement given as a title. Introduction gives a background based upon your review of literature, research question, and hypothesis statement. Then what is the statement of hypothesis, how it is tested, what kind of statistical analysis you're using to test the hypothesis, comparing between the groups, then significance of study. Literature review, about 2 pages or 1.5 pages, you can give literature review, giving the background information in detail. Here you given a very short introduction in the background, short one, about 1 or 2 paragraphs. Here you give detail about 1 to 2 pages. Methodology, experimental design, which Dr. Balkumar will be doing 5 and 6, the work schedule. So summarize the whole of my talk. So I think Dr. Deshpande will say, I'm on time. Still I got 5 minutes more at my watch because we started at 8:40 or I think. The fundamental principles of scientific research are the formulation of a problem. That is, the problem finding is an umbrella concept. Very big one, narrowing down. This umbrella I said, it's umbrella one, and then getting into this narrow one. The development of a study protocol or research plan and the description of what, why, and how of the protocol, literature search and review, study designs, statistical aspects, result analysis, and data interpretation. That is the main. Our initial vision for a study largely shapes our actions, approaches, analysis methods, what you do it in your laboratory methods, And then finally you have to interpret and what is the impact of your study. That is the need, societal impact. Very important. When we do webinar, what is the impact? This is what the Deshpande has already explained. What is the impact of our webinar on you people? So anything we do it, there should be some benefit accrued. Ultimately, the study should be suitable for publication. Very important, not to become just a showpiece in the library. You should be able to extract that particular work, publish an index journal, that is a quality publication, H-index journal. No plagiarized, hijacked journals. Very good journals. Again, there will be one more topic which we discuss about. Of course, our workshop goes for 2 days. We talk about starting from the cradle to the top, the final phase. So we cover from A to Z areas, from the way you start your research, the way you accomplish your goal, publish your work, and get a very nice position, industry or faculty. The scientific value or merit of published article is based on its own merits, not based on because just to publish doesn't mean that you are good, but what substance should be there, content should be there. The scientific enterprise requires a lot of thinking and imagination before one puts ink on the paper. Remember, the rest of the story. Intelligence, I already told you about this aspect with Someshwar Sathaka. That is, you'll be having all these things coming into a picture: intelligence, creativity, thinking, motivation, skill, environment, perseverance, learning strategies, perspiration, persistence, success. You know, Mankutimana Kagga, all of you must have read a lot of our people, got a lot of poets, our Master Venkatesh Iyengar, DBG, Kuvempu, Shivaram Karan, very literary figures are there, Dara Bendre. DBG's Mankutima Kagga, there are about more than 900 Kaggas. He says that Gharatattva Bandakke, Mana Dinaratri, Manasotu, Berundu Nenede. Very beautifully says Gharatattva Bandakke. For you are doing this work, and for that work you have to focus yourself. That is the one principle, any philosophy to solve it. Gharatattva Bandakke. Dinaratri, day and night you have to work. Manasotu, Berundu Nenede. We don't require anything. We don't want to think about anything else, but think only about this problem, how to solve it. So that is the one which say that we spend day and night to solve our problem. That is the one which project is should be written with the ink of creativity, intelligence, and hard work. All libraries, in my opinion, have this book because I must have donated more than a dozen books to our GCP library. Every department has got this book. Our library, I checked it. The pages have become so dirty that shows that people are using this book. But if you're not using it, please take time to read this book, which gives a fundamental aspect, published 2010, but the book is no longer available. But this duplicate copies are available. For all STARS Award recipients, we give a copy of the book. All students who get the AGCPA grants, they get a copy of this book for them to read. 600 pages one. It was 500 rupees. They escalated to 1,500 rupees. Still, it is not available. Even if you spend more money, you don't get it. And the next book, what we published last year, is the one which is the book. You people must have seen this book in the library. Very costly book, and it cost about $200. And I gave 2 copies to our GCP library, and Dhyanasagar University has got a few copies. Adi Chunchandgiry University has got few copies. I believe Manipal University authors also, they are written on the thesis, how to write thesis. Dr. Usha Naik and other people, Dr. Gurupa's leadership, and they also put a lot of copies. So many universities, they do have this copy of the book. If you do not have a copy of the book, please let me know that when I come in December during my visit, when I give a talk, I should be able to come up with this book. And I see that how you can get a copy of the book at a reduced price at zero cost to your library if you are genuinely interested. This is a book which gives a lot of fundamental things about publication, writing and publication, everything. Thank you very much for your attention to my talk. Thanks a lot.
Thank you so much, sir.
I stopped this sharing one. Okay, thank you.
Thank you so much for your time and a detailed presentation. Now Prakash will
So
introduce.
before we go go to the next session, are we going to take some questions?
There are no questions as such. If any student have questions, they can raise your hands. We can take one or two questions before going to next presentation.
Having charts. I don't know what are the questions are
Yeah,
there.
actually in
Yeah.
chat
Just
they are all providing their description from where they
but
are
no
coming
questions.
from. Yeah.
Yeah.
So I
No
believe
questions.
that yeah, somebody is able to ask question. Yes,
If
Ramya.
you have any questions, you can raise your hands.
Go ahead. Somebody is want to ask question. I think so. Dr. Ramya Bharati. I'm not able to hear. You have to be very high pitch because of distance. The speaker phone or come here to the speaker. Microphone.
No, I think she is not talking related to
Okay,
this.
otherwise, you can type it. If you think that you are not audible, type it. When Dr. Balkumar is giving a talk, I can always give the answer by typing it. I can do that one. I will check the chats next 1 hour. I can answer your queries. That's the best way. Dr. Deshpande, agree with me that
Yeah,
I will be able to answer the chats. In case
definitely,
if there
sir.
are questions, if there are any questions, then only I will chat. I will answer those chats. If there are any questions, please put the questions. Okay, I think I'll conclude and I'll give the way for others, other next speaker. Thank you.
Thank you so much, sir.
So, yeah. Thank you, sir. Our next speaker will be Professor Balakumar, and he will be speaking on essentials of experimental design and statistical analysis in a research protocol. Professor Balakumar is a professor and director research and training and publications, Office of Research and Development, Periyar Maniammala Institute of Science and Technology, Tanjavur, and he is also an adjunct professor of pharmacology, School of Pharmacy, Faculty of Health and Medical Sciences at Taylor's University, Malaysia. Professor Balakumar obtained his master's and PhD from in pharmacology, specializing in cardiovascular pharmacology from Department of Pharmaceutical Sciences and Drug Research, Punjab University. He was a professor of pharmacology at the College of Pharmacy, King Khalid University, Saudi Arabia, and a senior associate professor and head of the unit at the Faculty of Pharmacy, AMIST University, Malaysia. He was a visiting scientist at the University of Montreal, Canada in 2009. He's also an adjunct professor of pharmacology at Taylor's University, Malaysia. And he's also a consulting editor in pharmacological research, a Europe-based Elsevier journal with the impact factor of 9.3. Professor Balakumar's name is among the world's 2% of scientists list for almost four years in a row with the top 2% of scientists. Dr. Balakumar received many awards to his credentials. He is the recipient of a Star Award in 2015 for excellence in academic and research performance research performance at AMIST University, Malaysia. He also received Best Teacher Award from Periyar Maniammai Institute of Science and Technology in India. Additionally, he is also an author of 2 books. He's also in the editorial board of several journals, including WSCIE journals of international repute. He also has a publication in top tier journal Nature, along with the Dr. G. Jagdish. And he has published about over 130 scientific papers in the area of cardiovascular, diabetic, renal pharmacology, and toxicology. As of June 2024, his scientific papers have garnered about over 6,000 citations with the H-index of 42. So with that, we welcome Professor Balakumar. Professor Balakumar, floor is all yours. Thank you so much.
Thank you very much. Good evening to everyone. Am I audible?
Yes, sir.
So
Yes. So my
can you put your slides in presentation mode?
Okay.
Yeah, thank you, sir.
Is it okay? Am I audible?
Yes.
Yeah, I would like to convey my sincere gratitude to Dr. Deshpande, Dr. Prakash Bhagav, and Dr. Jagdish for having invited me to talk on the topic titled The Essentials of Experimental Design and Statistical Analysis in a Research Protocol. That is to be written by PG Pharmacology or Pharmaceutics or Chemistry or Cognitive students. My name is Dr. Balakumar. Currently, I'm working as a professor and director in the Office of Research and Development at Periyar Maniammai Institute of Science and Technology, deemed to be University, Tanjavur, Tamil Nadu, India. Well, science includes research. Problem statement. So, research can be defined as it is a original investigation in which initially the problem is identified. Then research questions are developed. And we need to do experiment and find a way to solve a problem. That is research. So research starts with the questions, research questions. Then watch, science. See, watch. You need to go through the literature search. You need to identify the information. You have to generate ideas. And you need to explain it. Once you explain it, you have to test it. Once you test it, you will get a results that you have to analyze them. Then once you analyze them using statistical analysis, you will reach to the conclusion. And that result should be reproducible. That makes research, and research is part of science. So dear participants, last year itself, more than 10,000 research papers were retracted. Why? It is because the plagiarism. Most of the students, not most of the students, some students knowingly or unknowingly, they do copying and pasting, the methodology part, the introduction part, and they steal figures, tables from others. End of the day, suppose if the paper is published, later on if it is identified and papers are being retracted. So you have to be very careful not to involve in any kind of practice of copying and pasting. And also, there is a thread for paper mill, means paying money for some authorship position and getting the paper without knowing the entire concept of the research. So this is also nowadays, enormously increasing, and all the students must be careful not to involve in such a malpractice of implicating yourself in involving paper mills. So for everything, there must be outcome. At the end of the presentation, the participants are expected to explain how to formulate a constructive methodology section and data analysis using appropriate statistical analysis. So dear students, the method section will be placed in two parts of the research manuscript. Two places. One is in abstract, other one is in the main body of the manuscript. In the abstract, there are 4 components. The first component in the abstract is your background and a research question. The second one is method. The third one is research, and fourth one is conclusion. And most cases, the second largest part of the abstract is methodology section. Means the reader should be able to understand what you have done and the superiority of the method, what you have performed, that should be in the abstract. Then followed by results and followed by conclusion. That's all. There is no discussion on the abstract. Then again, the second place where you have to concentrate elaborately to write methodology section is your main body of the manuscript. The component you can summarize as IMRAD. The acronym is IMRAD. Introduction, methods, results, and discussion. So introduction, as Dr. Jagdish said, why did you start? You need to come up with a prevalence data, research question, then hypotheses, then aims and objectives. So these are all things. Then at the end of the introduction, there must be aims and objective. Then followed by methods. What did you do? So methods should be written in past tense. We carried out, we analyzed, we performed. Sometime as an editor, we could see in the research paper, we will perform, we will analyze. If you will analyze, why you have submitted the paper, how come you have submitted the paper? This is question. So simple grammatical mistake will subject your paper for desk rejection. Desk rejection means your paper is rejected without being sent to the peer review for external reviews. So you have to be very careful. So methods, what did you do? Results, what did you find? Discussion, what does it all mean? So here in this presentation, particularly, I'm going to concentrate on the second part, methods. What did you do? And what kind of methodology structure that you have to follow. So this is what my presentation is intended to deliver to you. So I told the introduction in the IMRAD should be written in the present tense. For example, aliskiren is an renin inhibitor. You cannot write aliskiren was an renin inhibitor. So should be in present tense because introduction is written based on the existing literature. The earth is round. You cannot say earth was round. So it should be in present tense. Whereas methods are written in past tense. I told you, we carried out, we analyzed, we performed, we experimented, we tested. Then results, past tense and present tense. Past tense means we observe everything. Present tense means this figure shows, this table shows. So the legend can be given in the form of present tense. Discussion, most of the discussions are present and the present perfect tense. Okay, so well done. So let us come to the main point. So your research starts with the problem statement. The first line of your introduction, your protocol should have a problem statement. This is what you are going to do, going to solve the problem. For that, you are going to do experiments. So you have to come up with the background and the significance. Look at the protocol. These are the component of protocol that you must follow while you are submitting your proposal. So you have to come up with the first problem statement. Diabetes mellitus is a leading disorder. This much amount of people have been afflicted with the diabetes mellitus according to World Health Organization 2024 data. So such kind of the problem statement you need to mention. Then what is the background of your study and its significance, and what the specific target that you want to work in, and what are the existing knowledge available in that area through literature search. And you have to accordingly, you have to find out the niche area, the gap, which has not been addressed by anyone. This is what you are going to address in your study. Then what are the expected result that are associated with your work? That should be written, expected result, and how the anticipated results contribute to the field advancement. So this is you must mention in your protocol what are the results expected and how the anticipated results contribute to the field advancement, where the field where you are currently going to work, the research activities. Then you should state the hypothesis, as Dr. Jagdish told, and how the hypothesis is going to be tested. It is important to mention the hypothesis, and also important how the hypothesis is going to be tested. Then finally, research question. It is still unknown. It is not yet explored. It remains elusive. Therefore, the present study has been designed. So this is the last part of your introduction section, or the proposal, the protocol, aims and objectives. Then followed by experimental method, study design, and data analysis. That is not sufficient. You have to submit your work schedule. The first week of September, I'm going to do this one. Second week of September, third week of September, fourth week of September. That is known as Gantt chart. Then first week of October, second week of October, third week of October, fourth week of October. Then first week of November, I will complete my experimental part. Second week of November, I will perform the data analysis using statistics, some statistical software tool. Then third week, I will evaluate and compile the result and reach the conclusion. Then I will start writing the paper on the fourth week of November. The first week of December, I will submit the paper. So this is what the work schedule must be mentioned in the proposal. I could not see any work schedule in the proposals that are being submitted by the students. So the target and along with the work schedule is very important. Dear students, for methodology, now reaching to the key point, main point from this introduction, you have to follow the onion research model. I mean research onion. There are 7 parameters. First is philosophy. Second one is approach. Third one is strategy. Fourth one, choices. Fifth one, time horizons. The sixth one, techniques. And seventh one, procedures, data collection and data analysis. So these are the component of your methodology section. So let me explain one by one. The philosophy, the study of the fundamental nature of knowledge, the reality, the existence, how you are going to test the hypothesis, what kind of study yours. It is a case control study, what kind of study. Then approaches, what will be your approaches, whether your study is a inductive or deductive. Inductive is a qualitative study on assumption and you are reaching conclusion through the experiment based on deductive. So inductive or deductive. Then what kind of strategy that you are going to follow, whether it is a survey study or experimental study or case control study, what kind of study it is. And what are the choices, what kind of methods, whether you are going to follow mono methods, multiple methods, or mixed methods. For example, you are going to analyze the kidney section architecture of the rat. There are different kind of stain, and you want to see the structural abnormalities in the cortex, renal cortex. So I'm going to use hematoxylin eosin staining, and also I'm going to use periodic acid chip staining and Mason trichrome staining. So multiple method, sometimes mixed method. For example, you are going to do tunnel staining for apoptosis or some kind of fluorescent assay, or acridine orange test for apoptotic measurement. So mixed methods I'm going to do. So these all things very clearly, elaborately, and with the advanced technique. Not simply I'm going to do Ridding model, analgesic activity, antioxidant activity. So these are the activities. If you do, you will unlikely publish your paper in a journal of international repute. Like Web of Science, Science Citation Index expanded, Q1 or Q2 or Q3 or Q4. And your target should be, at the end of the day, publishing your paper in a well-reputed, globally recognized databases like Web of Science, Science Citation Index expanded. So you have to come up with advanced techniques. The next time horizons, like your study is a cross-sectional or longitudinal, even in the title itself, a cross-sectional study, an observational study, a case control study, a longitudinal study, that you have to mention. Very clearly you have to mention. What does it mean cross-sectional? The data will be collected at one point of the time. That's all. For example, you are doing an experiment today after the ethical approval and you are sacrificing the animal at day 28. And at the day 28, you are collecting the samples and everything and you are analyzing them for experiments. So the data is collected at one point in time. That is cross-sectional. Longitudinal means the data are collected repetitively. Day 1, day 3, day 10, day 30. For example, you are providing some pellet to the rat and body weight is measured. What about the day 0, the day 5, day 10, day 20, day 40? So you are collecting the data. That is longitudinal study. So finally, once the data are completed, you have to collect the data and subject them into descriptive analysis. Mean plus or minus standard deviation, you have to express the result. The measures of central tendency you must know. Then you must subject them to inferential statistics. And you have to confirm what kind of data you have collected, whether it is a nominal or ordinal or interval or ratio data. Accordingly, if it is a interval or ratio data, whether the data are following uniformity or not, the distribution is normal or not. If they are normally distributed, you have to go for parametric test. On the other hand, if they are non-normally distributed, you have to go to non-parametric test. What are those parametric tests? How many groups of you have, whether it is a true group you are comparing on the same subject, then paid t-test. If it's a different subject, 2 group, unpaid t-test. So these all things that you have to mention in the proposal protocol before you submit it for approval to the specific body in your college. So this research philosophy mainly have positivism, interpretivism, and pragmatism. Interpretivism is inductive. So interpretivism is inductive. So inductive is a qualitative, like case control study. We don't know. It might be because of this one. This drug causes this particular side effect. It is because of this one, because it is not experimentally proven. It is a inductive. When you are able to prove it experimentally, then that is detective positivism. Interpretivism is inductive, whereas positivism is deductive, and pragmatism is both inductive and deductive. That is another kind of term, that is realism. You want to explore the issues of real life learning understandings. So these all things you have to mention in the research methodology. Like Dr. Jagdish said, inductive and deductive. For example, you are saying that every dog I meet is friendly. So what you will hypothesize? Most dogs are friendly. So this is your hypothesis, but you have not experimentally proved. For example, if it is a deductive, all mammals have backbone. Humans are mammals. So humans have backbone. So you have some evidence. So based on that one, you are reaching the conclusion that is deductive. And while you are structuring the methodology section, for example, you are going to measure protein estimation. You are going to do protein estimation by low-risk method. Keep the reference very clearly. The protein was estimated, bracket, low-risk method. You have to cite the appropriate reference. Otherwise, while you are submitting your paper, there is a machine learning tool available with the publisher. For example, Springer uses X-Tiles and elsewhere uses Mars, machine learning referencing system. So this machine learning software will identify how many references that you have cited are valid and how many are invalid. For example, you are citing 30 references, out of 30 all are invalid, your paper will not go through, will be test rejected. So you have to cite appropriate reference, very meticulous references. Okay, coming to the point, these are the 5 components that you have to include while you are structuring your materials and methods section. The starting point should be ethical approval of your experimental protocol. The protocol that has been performed in the present study has been approved by institutional ethics committee, along with the approval number, date, and everything. And why, if you are using animal study or human study, and how you have treated the animals separately in the cage, day and night cycle, free supply of water and food. So everything you have to clearly mention the first paragraph, ethical approval of your experimental protocol. If your experimental protocol is not approved by your ethics committee, means you are unlikely to do experiments. First, you get approval from ethical committee, and that paragraph should be the starting paragraph of your materials and methods section. Then the second one, the experimental protocol. 7 groups were employed in the present study, and each group consists of 6 to 10 animals. 6 animals, 7 rats, 5 rats, whatever it may be, that you have to mention what is group 1, what is group 2, group 3, group 4, group 5, group 6. Either you can present in the form of schematic diagram or you can write in the paragraph, does not matter. But the second section of your materials method should be experimental protocol with the experimental group. Group 1, group 2, group 3, group 4, group 5, whatsoever it may be. Then third part should be your methodology and parameters. What kind of parameters that you are going to do? Estimation of interleukin-1, Western blotting, histopathological analysis, mono method, multiple method, mixed method. So whatever the method, and make sure that the method what you are going to perform is an advanced technique, advanced method. For example, TN of alpha you can measure through enzyme linked also assay also, and through Western blot also. Which one is better? Western blot. So where we can perform this one? What will be the cost? How I can perform this particular methodology? So you have to try to come up with the advanced technique rather than the conventional technique. So the second part of that one, I mean third part is methodology and the parameter. For example, simple in vitro antioxidant assay also you can do it. You can do fluorescent assay for reactive oxygen species also you can do it. So which one is better? The fluorescent assay. You can simply measure apoptosis through kit, and you can do tunnel staining also for measuring apoptosis or acridium orange. So such kind of advanced techniques, if you follow, you will likely open the door of processing your paper smoothly for publication in the Journal of International Repute. That is none other than Web of Science, Science Citation Index expanded Q1 or Q2 or Q3 or Q4 journal according to the quality of your paper. Then fourth section of the materials method should be statistical analysis. Keep in your mind, after attending this particular webinar, you should never express your data as mean plus or minus standard error mean. Standard error mean is not an index of variation. Standard deviation is the index of variation from the mean. So you have to express your data as mean plus or minus standard deviation. And all the data are expressed as mean standard deviation. In between two groups, we have employed this test for analysis, and more than two groups, we have employed that test followed by post-hoc test. And we confirmed the normal distribution of the data through Kolmogorov-Smirnov test. So what kind of test you have applied to initially confirm whether the data is uniformly distributed or not? So these all things you have to mention in statistical analysis. Finally, the fifth part is drugs and chemical. All the chemicals employed in the present study are of analytical grade, not a laboratory grade. So you have to purchase the chemicals from a leading suppliers like Sigma and others. So these are the component of your materials and methods section. Now see the group. Look at this one. Effect of compound X in gentamicin-induced nephrotoxicity in the rat or in cell lines. Maybe proximal tubular rat, proximal tubular cell lines. Whatsoever it may be. The group. See the protocol. I have come up with a 7 group. If it is a rat study, I might request n equal to 6 because there is not too much mortality. So I can save the animal throughout my experiment. If the mortality is high, you have to increase the number. But still, you have to get ethical permission after justifying that why you are asking more than 6 rats or mice per group. So group 1 always normal control. So for example, you are using a carboxymethyl cellulose as a suspending agent, for example. So that you can give to normal group so that the drug has effect, not the CMC. So kind of negative control. Then group 2, the test drug. I mean, most of the people are not following this one. What test drug you are going to evaluate, that should be given to the normal rat, normal animal. That is per se effect. Whether the drug itself has any per se effect or not. So if you are giving the drug to the disease animal, definitely it may or it may not ameliorate the disease. But what is the effect of test drug in the normal animal? That is per se effect. So, for example, you are going to use 3 dose means the high dose should be given to the normal rat. That is per se effect. The third group, only the disease control group. The disease, for example, I am giving here gentamicin to induce nephrotoxicity. Now, fourth group, treatment group, the test drug 1 or test drug at low dose, then test drug as intermediate dose and high dose. So the doses you have to calculate according to the toxicity studies and other body surface and other things. Human dose you can convert, easily you can convert like that. And in these all groups, I will give drug pre-treatment along with the gentamicin. Then finally the standard compound. If any nephroprotective compound is there, that can be used as a standard compound. So these 7 groups will make more sense. So here the positive control standard group. Disease control is a gentamicin group. The negative control, normally it is a placebo effect, that is negative control. Many students, they confuse what is negative control. So you can simply evaluate the effect of solvent that is in normal control group. No need to put extra group. So these are the minimum groups. And try to use 3 doses of test drug. Don't evaluate only 2 doses. 3 doses. So the dose response curve is always effective. It is the 3 doses. Low dose, middle dose, and higher dose. So parameter, see, for this study, what parameter? MTT assay for cell viability, if it is in vitro assay. Then I want to evaluate the oxidative stress, RAS fluorescent assay. Then I want to see the apoptotic cell death and ameliorative effect of drug on apoptotic cell death. Ethidium bromide, acridine orange assay. So it is a costly one, but still it is a better one to evaluate apoptosis. Then I want to go for matrix metalloprognosis estimation. Then I want to go for quantitative reverse transcriptase polymerase chain reaction. Because this vascular endothelial growth factor, osteoponin, are reported to be modulated during gentamicin nephrotoxicity. I want to measure them. I want to go for a quantitative reverse transcriptase polymerase chain reaction. Then a simple antioxidant effect, like superoxide dismutase and glutathione peroxidase. Then I want to carry out some Western blotting study to measure the protein expression directly. Then finally, some anti-inflammatory effect I want to check. So I want to use enzyme-linked immunosorbent assay, like interleukin 1 or 6 or interleukin 18, or anti-inflammatory cytokines like interleukin 10. So these are the attractive parameters that you have to make it, not simply simple assay like antioxidant, this one, anti-inflammatory, like that. So the way you are coming up with the protocol will make a door, will make a door opening for you to easily, smoothly publish your paper in a Journal of International Repute. So coming to the analytical part, so the statistics, the data. So there are inferential and descriptive statistics. Descriptive, I told you are describing, and inferential, you are testing the data. So descriptive and inferential statistics are two division of statistics. If you want to summarize the data according to the mean standard deviation, so these all are coming under descriptive. If you want to analyze the data, whether this group is statistically significant than this group, so that is inferential statistics. So this is what the descriptive statistics include: central tendency, the dispersion. Dispersion, I told you, it's not a standard error mean, it is a standard deviation. Then skewness. Skewness means there is no normal distribution. There may be a positive skewness or negative skewness. Both are not normally distributed. Both are non-normally distributed. So you cannot use parametric test. You have to use non-parametric test. So kurtosis, there is a tailedness, kurtosis, that is different from skewness, and many people they confuse. Kurtosis is different, skewness is different. In both way, if the data follows skewed, then it is not normally distributed. If you go to Agerson-Pearson test or Kolmogorov-Similar test, and you will get normality test failed. So you cannot use parametric test. In that case, you have to use non-parametric test. So you have to make it. See, positive kurtosis means tailedness, negative kurtosis. Okay, so let me leave this one. So data, there are 4 kinds of data. Mostly the paramedical, biomedical sciences data are coming under 4 categories: nominal, ordinal, interval, ratio. What does it mean? See the data types. You can classify into 2 major classes: categorical, numerical. Categorical data are again 2 types: nominal, ordinal. Numerical data, interval, ratio. Most of the categorical data you have to use non-parametric test, and most of the numerical data you have to use parametric test if they are uniformly followed. So everything depending upon the uniform distribution. Nominal data. For example, in this particular webinar, how many boys and how many girls are attending. So such kind of data can be only numbered, cannot be quantified. It can be only numbered. For example, out of this 150 participants, 80 are boys, okay, 70 are girls. So boys 80, girls 70. What about their knowledge after this one? So the knowledge is this much. The amount of grasping the information from the seminar is this much percentage. So you have to come up with a contingency table, 2 into 3 table, 2 into 2 table, 3 into 2 table. So accordingly, you have to apply chi-square test. So nominal data always involves Fisher test or chi-square test. That is different. For example, 11th grade student, 12th grade student, whether they have cell phone or not. How many 11th grade students have cell phone? How many 12th grade students have cell phone? How many do not have it? So this is 2 into 2 contingency table. So if you apply chi-square test, you will come to know whether there is a significant difference or not. Then ordinal data. Again, ordinal data cannot be measured. It is a categorical data. It is a categorical data. For example, mild pain, moderate pain, severe pain. For mild pain, you are giving 1+. Moderate pain, ++. Severe pain, +++. Extremely severe pain, ++++. So some kind of qualitative data. So for that one, again, you can make it into the numerical. For example, see the Likert scale. Strongly agree, disagree, neutral, agree, strongly agree. So for strongly disagree, you give -1. Disagree, 0. Neutral, 1. Agree, 2. Strongly agree, 3 points. Make a, see, the qualitative now is count now. Now we can count the things. But still we cannot measure the things. So such kind of things are Likert scales are coming under ordinal data. For example, you like this webinar or not? Some students may say yes, some students may say maybe, some students may say no. So accordingly, we have to give some mark and see whether significant difference or not between these three categories. Okay, so the data type may be categorical or numerical. Categorical comes under, I mean, the two further division, nominal and ordinal. For example, numerical, interval, and ratio. For example, interval. Interval data are coming under numerical, not a categorical, because we can measure it. What is the temperature of this room? We can measure it. But the difference between interval and ratio is interval has true 0, ratio data is not having true 0. For example, 10 degrees Celsius, 5 degrees Celsius, now cooling is there. 0 degrees Celsius, very cooling, minus. So 0 has a value. For example, 0, 0 is to 0, 0. It means 12 o'clock. 0 is a value. But whereas ratio data, there is no natural 0. For example, I want to measure your weight. Weight may be 20 kilo or 30 kilo or 40 kilo or 60 kilo or baby maybe 2 kilo. There won't be any 0 kilo. So such kind of data are ratio data. So both interval and ratio data are coming under numerical. Mostly they will be normally distributed. If they are normally distributed, you have to use parametric test, otherwise non-parametric test. Okay, students. So variable may be categorical or numerical. Categorical, I told, nominal, ordinal. You forget it. You forget it. Now, numerical. Numerical can be discrete or continuous. Discrete means a round number. Continuous means decimal point. For example, how many times you visited a dentist? 5 times or 7 times per week. You cannot say 7.4 times. So that is not a discrete. Discrete means the round value. How many people they have died in the hospital because of coronavirus infection? 10 people. You cannot say 10.2. So such kind of data are discrete data. For example, what is your height? 172.22 millimeter or centimeter. What is your weight? 57.3 kilogram. So the decimal points are coming. That is known as continuous data. So this kind of data you have to understand first before you apply for the test. Now see, this is a very complex one. Data are categorical or quantitative. The categorical data is qualitative. Again, they divide into discrete. Discrete, again, nominal, ordinal. Then quantitative, continuous. Again, interval, ratio. So these all things I have explained to you. I hope you have understood. So you must understand the difference between standard deviation and standard error. Standard deviation is that quantifies the variation. I mean, how much the data is varied. For example, you are measuring the glucose level of all the students I am going to measure. So starting 90, 91, 110. So the variation is 90 to 110. So that is the variation. The mean is 100. So that is standard deviation. Standard deviation divided by root n is standard error mean. So you cannot express your data with the standard error mean. So always you should express your data as a standard deviation. That is the variation, the true variation. So most of the protocol proposal, I could see that data will be expressed as mean, standard error mean. It is wrong. You cannot publish in a good journal. It will be right away rejected. Okay, fine. So there are two distributions. I told normal distribution. In normal distribution, there is no skewness. No skewness. The data are normally distributed. The minimum 3 standard deviation. First standard deviation, second, third. Second standard deviation is the 95%. I mean the confidence interval, 1.96. Then non-normally distributed means skewed from left side or right side. Any side it may be skewed. So such kind of data should be analyzed using non-parametric test. So if your measurement scale is nominal or ordinal, you might need to use non-parametric test. For nominal, only chi-square or Fisher test. If it is ordinal, if they are not uniformly distributed, then you can go for non-parametric test. One student asked me a question, sir, if it is ordinal data, but if it is uniformly distributed, then you can go for a parametric test. That's not a matter, but that happens in rare case. If you are using an interval or ratio scale, mostly they are not normally distributed. How to confirm? you have to confirm by Kolmogorov seminar test. Anderson Pearson test. You have to use it. So these tests are very much useful. Automatically, you will find out whether your data is normally distributed or non-normally distributed. See here, the level of nominal, ordinal, mostly non-parametric. They are qualitative data, cannot be cannot be measured, only can be counted. Interval ratio data. So this is the confidence interval 95% means 95% of the time the data lies within the particular standard deviation, variation. This is confidence interval. So 0.025, this time 0.025, that is 0.5, 0.05. 0.05 means 5%. Means 5 divided by 10, 0.5. 5 divided by 100, 0.05. That's all. That is the p-value. Okay, fine. So this is the confidence interval we can measure. Sample mean plus or minus 1.9, which is the second standard deviation into standard error mean. Okay. So there may be some time error, type 1 error, type 2 error. Type 1 error is a false positive, type 2 error is a false negative. The error may be made by incorrectly rejecting the null hypothesis, that is false positive, or incorrectly accepting the null hypothesis, that is false negative. This may be possible because of you are expressing the data with the standard error mean, or you have 3 groups. First 2 group you are comparing, then second, third group you are comparing by t-test. No, you should use ANOVA. So such kind of things, if you properly follow the statistical analysis, you can come from this problem, that is false positive and false negative. Okay, fine. So this is the chart, the diagram, the figure we have published in one of the journal of pharmacology. The 2 groups both are paid. Okay, now clearly paid group means you are measuring my blood sugar level. You are measuring my blood sugar level now, for example. Okay. So is it okay? You are measuring my blood sugar level now. Now I am eating 1 kilo jalebi. You are measuring my blood sugar. There will be variation. So from the same subject, you want to see the difference before taking sweet and after taking sweet, or 1 hour. So paid group. So if it is a parametric data, you have to use paid t-test. If it is a non-parametric data, you have to use Wilcoxon sign rank test. If it is unpaid, for example, 50 students are here, 25 control, 25 are subjected for some sweet. After 1 hour, we want to see the blood sugar level. Okay, so different subject. So if they are uniformly distributed, unpaid t-test. If they are not uniformly distributed, non-parametric, then Mann-Whitney test. That's all about the 2 group. What about more than 2 group? If they have 3 or more group, if they are matched group, day 1, day 3, day 5, such kind of matched group. If they are uniformly distributed, then repeated measures of ANOVA. If not, Friedman test followed by Dunn test. If it is a repeated measures of ANOVA, you have to also involve students' Niemann-Kuhls test for the data which are normally distributed parametric matched group. So if they are unmatched group, for example, normal control, gentamicin control, treatment 1, treatment 2, treatment 3, all are unmatched. If they are normally distributed, one-way ANOVA, followed by Tuckey's multiple comparison test, or Bone-Ferroni's correction test, or Duncan multiple range test, or Dunnett's test, or Niemann-Kuhls test, also here you can use it. Okay, if they are not normally distributed, then only one test, Cruxel-Wallis test, followed by Dunn test. Dunn test is the only post-hoc test that can be used for matched group and unmatched group. So this diagram is freely available. You can type Dr. Jagdish sir name or my name. You can enjoy reading this paper early morning. So this 6 component you have to enter into your protocol. How much should be the sample size? Are the data uniformly distributed or not? Which test should I apply? What post-hoc test should I apply? Is there any correlation between variable? For example, 2 variable, the height of the person and the size of the chapel. Height, if height increases, chapel size increases. If it is a parametric test, apply Pearson correlation. If not, Spearman correlation. Then what software you have to, whether it is a GraphPad software you are using, GraphPad Prism or SPSS, you mention it. You mention it. So you see the well-written paper should go through several gatekeeper like journal administrative staff, receiving editor, chief, then associate editor, then reviewer 2 to 8. Everyone should be convinced by your methodology section. Then only your paper can go through it. Your students, you should avoid publishing, like Dr. Jagdish told, predatory and non-index journals. You should avoid. You should always publish in a above science citation index expanded journal, and also you should improve your citation index and H index. That can happen only by publishing in a quality journals. So your paper acceptances will increase if your paper describes field advancing the research, if it is expertly written and formatted, if it speaks in a clear and concise manner, and it follows the ethics and free from any scientific misconduct. I told more than 10,000 papers were retracted last year itself. And this year we don't know. It is all because of misconduct. So you should never be the victim of any misconduct. So number of papers, you can type our name, number of reports, papers are available in the internet. You can enjoy reading this paper. So whatever we have presented, that may or may not be the part of these papers. So this is a recent one. This is recently we have published Journal of Ayurveda and Integrative Medicine having impact factor 1.6. So Dr. Jagdish told the book you should follow. Although it is a costly book, it is given in some libraries, so you can go and refer and possibly take a photo state and keep reading this book to become a wonderful researcher. Initially, your paper may be rejected. You'll keep on doing. If you do the good research, definitely one day you can become a good researcher. Thank you very much for the opportunity. The organizers.
Thank you so much, sir.
I will be pleased to address if there is any question, myself or Dr. Jagdish sir. So
Yeah,
the audience have any questions.
Any attendees, please, if you have any questions, please raise your hands for both presenters.
So the time is fixed at 8 o'clock. I finished by 7:58.
Yeah. I think
Thank you so much, sir.
we are on time. Still,
This
we are
all
not about 5
training, this
minutes
all
more.
training were provided to me by Dr. Jagdish sir. You must be time follower. Means if you half an hour given, we should follow only half an hour, not
Yeah,
even 31 minutes.
he is always on time.
That is true, sir.
So, any questions from the participants?
I answered most of the queries, whatever has been posed by attendees, and especially people were concerned about the dose selection. I know very well it's a very critical dose selection. As I said earlier, people use too low dose or too high dose without going through the literature. For most of the drugs, doses are available. And in spite of it, people, I don't know why they make mistake, especially when you use nano-formulated or selenium or something, whatever you do site-specific delivery, you should be careful in selecting a particular dose. And about extracts, people may use a different kind of extracts. Somebody is used with ethanol, now the people switch to different kind of extract. Definitely, the dose will be different. It cannot be same because the plant extract from different solvents will give a different constituents, and also toxicity may decrease or increase. So everything cannot be same. Again, the dose differs. If you don't find the exact match in the literature, then you must carry out the toxicity studies 1 to 7 days based upon the OECD guidelines. Dr. Naik also asked the question that is, and also other two more people, that is, how do you select the dose in work? Because I come across many students making mistakes, and also in particular about that plant extracts, you should be very judicious. In case if it's a very new plant, then you must carry out the toxicity test, that is 1 to 3, but don't kill the animal. Don't use what is called as the Litchfield-Wilcoxon method. That is nothing but a LD50. LD50 should not be used. So think about where you can give a dose. The animal should not die, but produce severe adverse effects. That is the maximum dose. That is based upon the OECD guidelines. We will be able to have one more talk on this one, either online before November, because I will be in India from December through January end and February first week. Myself and Dr. Balkumar and our big team is going to have a series of workshops at several places. If any one of your institutes is interested in having workshops or symposium for half a day to 2 days, let me know in advance so that we can plan for it and fit in our schedule. Already our schedule is getting filled up for December and January talks. So please let you know that some people ask questions regarding the pharmacy practice and Form D program, their project work. I did discuss with this what different universities, and we can also have some projects, what kind of projects these people can do. And sometimes they do either single or in group. I have no idea why they do in the group wise and what could be the community benefit. At AIGCPA, we have sponsored this community pharmacy scholarship for last 23 years. So for the benefit. So similar type of programs can be taken up by pharmacy practice people or Form D people. So we can have some ideas. Let us know how we can share our values with you people. Thank you.
Thank you so much, sir. Prakash?
Yeah, thank you so much, sir, both Dr. Jagdish and Professor Balakumar for this wonderful session. So before we conclude, we at AIGCPA would like to thank Dr. Jagdish for his valuable, informative and thought-provoking session today. The session was very well designed, scientifically designed, and putting a lot of efforts and thoughts in it. So we hope that our scientific community benefit from this session. And we appreciate, sir, we appreciate your ever readiness to help the research community in the state of Karnataka and in general and GCP in particular. Thank you so much, sir.
Thank you.
And our heartfelt thanks to Professor Balakumar for your valuable time for in designing your presentation, and your session was very informative from the protocol design point. And yeah, so we hope that your session was very well taken. Thank you so much. So we would like to thank Dr. Shiv Shankar for extending this platform to us. So because of you, we could have this, we could host this session for the scientific community in the state of Karnataka. Thank you, Shiv Shankar. We would like to thank all our AIGCPA office bearers for helping facilitating this session. And finally, all the participants who joined us from all over the places in India. We hope that this session is useful for you, and this session inspires you for better designing your protocols, your manuscripts, research grants, etc. So we look forward for your continued attendance for our webinars. Yeah, if you have any additional specific questions, please email us. Srikant, did you put our email address there?
Yeah, now also I'll put again. And if you have any feedback, any future webinars you would like to see from us, please let us know so that we'll make sure you get that resource in your hand at any time. So this is our scientific philanthropy we are planning to have from AIGCPA so that everyone should get research related exposure. That's what our Dr. G. Jagdish, as a founding member of AIGCPA, is the views, so that we try to make sure you get all the resources for your research related topics.
Yeah,
I am providing again our email ID so that you can shoot email related to your feedback or any help. Let us know. Thank you so much.
Thank you so much.
Dr. Deshpande, and thank you, Dr. Prakash. It is really a noble initiative that you are showing a right way for the student to walk into the field of research. Thank you for your steps and initiatives.
Thank you so much, sir, for kind words. And we'll now we'll conclude our webinar.
Thank you.
So good night to all our fellow Indian students in India, as well as to Professor Balkumar sir. And have a good day, Jagdish sir. See you. some other day in other webinar time. Thank you.
Thank you so much.
Thank you, everyone.
Bye.
Thank you. Bye now.